thank you so much for that introduction. And I was just thinking, Mariana, as you were saying, supporting early career researchers. And so I feel particularly fortunate to be included in this amazing lineup that you have for this seminar series, considering myself an early career researcher. So thank you so much for the invitation for coming to speak. Thank you all for adjusting for the time to work with Australian time zones and see how we can marry that up. And thank you for those who are tuning into the recording later. I understand, unfortunately, we can't get right around the world, so thank you for coming and listening as well. So as Mariana mentioned, I'm a conservation geneticist and research scientist at CSIRO, and I'm really endlessly curious about how we can understand evolution and particularly adaptation to environment, how that has shaped genetic variation and how we can use that for conservation. So as Marianne mentioned, my work has been predominantly on plants, but more recently on trees. And that's what I'm going to focus on today. So it's customary in Australia now to acknowledge that we are working and living on unceded territories of traditional owners. So I would just like to begin by acknowledging the Muanina people as the traditional owners of the land that I am presenting from today, and pay my respects to their elders past, present and emerging. So this year, I'm sure as most of you are aware, is the first year of the UN Decade on Ecosystem Restoration, which is really recognising the growing recognition of the importance of healthy ecosystems for our planet as well as our society more broadly. And indeed, even just last week in Glasgow, we saw over 100 leaders commit to not only halt, but reverse forest loss and degradation by 2030. So there's this really growing global interest in restoring healthy ecosystems more generally. But one of the challenges that we have is that we're working in a really dynamic system. So we know from climate projections and ecological modelling that there's significant environmental change already has occurred or is predicted to occur in the future. And from a conservation and restoration point of view, it means we're no longer just simply preserving the presence of species, but rather we're needing to support their capacity to adapt and change. And genetic variation is naturally a really important component of this, enabling populations to evolve. And indeed, there's even increasing recognition now of that importance of genetic variation for conservation, coming up as a milestone in the draft Global Biodiversity Framework that is currently out. And Mike Burford gave a really great introduction to conservation genetics last week that I highly recommend checking out if you haven't seen it already. So today I'm going to be talking about how we can use genomics to help these restoration and conservation efforts to build that resilience to change. So that for these endeavours, we're trying to ensure successful outcomes, both in the short, but also that really long term. And I'm going to go through a number of studies from work from both my PhD and my postdoc to date. The first of those are going to be focused on restoration and how we can use genomics both to assess restoration efforts as well as inform future restoration efforts. The second component, I'm going to look at how we can potentially use genomics to predict vulnerability. Now, this could be in natural systems or it could be in restored systems. But how can we apply some of the knowledge we're gaining about adaptive variation to understand the potential resilience or vulnerability of populations to future change? I'll also put a little preface at the start of this restoration component that even though I'm talking about restoration, there are a lot of mirrors in the forestry industry in terms of thinking about that longevity of populations and plantings that are put in place. So part of the aim of ecological restoration is really to create these long-term self-sustaining populations that are going to be able to recruit and adapt to conditions both now and into the future. And so with this increased focus that we're having now through these increased targets, through this UN Decade on Ecosystem Restoration, and all these additional efforts, how can we make sure we're maximising the outcomes of those for resilience into the future? For restoration plantings, these have traditionally sourced material using what's called local provenancing. So making that assumption that that local material is most adapted to the local conditions and therefore most fit. And we see this in a nice example of Eucalyptus gunnii from Tasmania, where I'm calling in from, where we see the local provenance, the open circles, have higher survival than the non-local provenance, those closed circles. Whilst local provenancing can be really, really effective, what it doesn't recognise is the dynamic nature of the system that we're working in, and especially for long-lived species such as trees, climate change can result in a mismatch between the climatic optimum that those populations are adapted to and the current climate at any particular site, such that the local trees may no longer be most appropriate and most adapted to local conditions. And indeed, going back to the example of Eucalyptus gunnii in Tasmania, we see that whilst for that first period of time, that first sort of 20 years, the local provenance was better in this location, over that period, we've seen increases in temperature, decreases in rainfall, to the point where the local provenance is no longer best. It's that non-local provenance from those warmer conditions that actually now has the higher fitness at that site. So given these changes, there have been quite a number of new restoration strategies aimed at incorporating evolutionary thinking and genetic variation into restoration plantings to enhance resilience. I'll come back to these in a moment and discuss them in a little bit more detail, because what I wanted to start with was how we can use genomics to assess what has currently been done in restoration. A little bit of an introduction of the systems that I'm predominantly going to be talking about that I work in, and those are eucalypt woodlands in Australia. So on this map, the lighter yellow colour is the current extent of eucalypt woodlands, and the dark blue colour is the woodlands that have been cleared since colonisation of Australia. So we only have about 34% of our eucalypt woodlands remaining, and these account for 42% of the total cleared land in Australia. This primarily, certainly for eucalypt woodlands, is due to the fact that they overlap with really productive landscapes and so have been extensively cleared for agriculture and pastoral use. The primary species I'm going to be focused on is Eucalyptus microcarpa, or grey box. This grey box grassy woodland, shown in the beautiful picture in the background, is a federally listed endangered ecological community here, with only 10 to 15% of its original extent remaining. It's widely used in restoration across its distribution in southeastern Australia to try and mitigate those losses. It also occurs across a number of wide environmental gradients. So it makes a really lovely case study to look at both genetic variation and restoration, and how genomics can help understand both. So looking firstly at how well have we done to date in terms of capturing genetic diversity in our revegetation sites. So this was 7 locations where I sampled both natural sites and revegetation sites, and just comparing diversity within those sites. So this is expected heterozygosity. What we can see overall is that the revegetation sites, the crosses, generally have lower diversity than the natural sites. However, this varies between sites. You can see there are a couple of locations there, the Rock and Crowther, which have really similar levels of diversity in their revegetation sites. And even for those sites where there was lower diversity, that was fairly low. It was only about 7% lower. So there wasn't a really huge decrease in the revegetation. This mix of patterns as well, to digress very slightly, also led me to think, can we look in the literature and how well and how common is this pattern more broadly? And so with a number of colleagues, we did a review of the literature, looking at over 40 studies that had measured genetic variation in both revegetation and natural sites. And similarly, we find this really diverse range of results. So we had almost a 50-50 split where revegetation either had higher or lower diversity than the natural sites. And this was for all 4 measures of genetic diversity that we had there. So number of alleles, allelic richness, expected heterozygosity, and percentage of polymorphic loci. So we have quite variable results in how well we capture diversity in revegetation. Coming back to that grey box example, Looking then beyond and looking at diversity and differentiation between sites, we wanted to look at how well were landscape patterns of genetic variation captured in the revegetation sites, and possibly any significant differences across those, such as adaptive variation. In the dots here, we can see that between our natural sites, we see a real pattern of isolation by distance, with gradually increasing differentiation between sites with geographic distance, which is what we would expect for this species. There are no real geographic barriers across its distribution, and prior to colonisation, it would have been really quite widespread across that range, and so there would have been historically quite good gene flow. If we look in at the restoration sites in the crosses, what we see is that they are more different at any given geographic distance than the natural sites. So it's suggesting they're more different from each other, and those patterns that we saw between the natural sites aren't being recaptured in our revegetation sites. So it's suggesting that there's some change in the variation that we're seeing in the revegetation, and that those landscape patterns and potentially any adaptive patterns that might be in there are potentially not being captured. These couple of metrics of expected heterozygosity, diversity, and differentiation have been commonly measured and can be measured with traditional older marker systems as well, such as microsatellites. Given I had genomic data, so this was reduced representation genomic data, but still a good genomic data set of about 13,000 SNPs, I wanted to see if we could really dig into some of these differences further to understand them and see if we could untangle what was happening here. So coming back to that diversity with insights, I wanted to try and look at, just as a first pass, what is that diversity like when we look across the genome? So for Eucalyptus, we have a really, really good reference genome that we can use. So I could split those SNPs that I had across the 11 eucalypt chromosomes. So this figure on the right is just an alternative representation of the figure on the left. It's showing the difference in diversity in the revegetation site compared to that natural site, with those red bars showing a decrease. So you can see most of the sites have a decrease. The rock and Crowther have similar levels of diversity. If we look across all the 11 chromosomes though, what we see is that there are really uneven changes in diversity across the genome. So that overall genome-wide summary of diversity isn't necessarily representing all the changes that we're seeing, even when we look at it at this sort of coarse chromosome level. So we're seeing some chromosomes that have either more or less loss of diversity, but we also even see some chromosomes that have higher diversity in our revegetation sites than our natural sites. So certainly this goes some way to explaining those differences between having similar levels of diversity but having greater differentiation, is that the patterns of diversity across the genome are varying quite greatly. So bringing this little bit together, what we can say is that we have really good potential to capture good genetic diversity in revegetation sites. But this can be really quite different when we look at what's captured right across the genome. So as we get better understandings of what genomic regions may be important, a better understanding of adaptive variation, we can possibly tease apart some of those genome-wide metrics and start honing in on particular areas or particular regions that we might want to focus on that may be associated with resilience in revegetation. And certainly, both from the grey box work and from the literature review that we did, one of the things that really came out was, despite the fact we had these varying results, we had very little metadata to understand what was driving those differences. And so very little capacity to make recommendations about what could be done to increase diversity or increase particular aspects of diversity in revegetation. So certainly as we're going forward with these increased efforts towards restoration, as well as trialling these new alternative strategies, making sure we capture that data is going to be a real key to being able to come back and really effectively monitor and therefore provide information back to management of how we can continually improve restoration. So that's a little piece on how we can use genomics to really monitor revegetation in much finer detail than we potentially have in the past. I'll now move to how we can think about using it to guide future efforts. So coming back to these alternative seed sourcing strategies. So these are aiming to enhance evolutionary potential by including genetic variation. And there's two key components of genetics that are being considered. One is thinking about just increasing genetic variation and diversity generally. So these are the two strategies on the left, sampling widely across the distribution or mimicking patterns of gene flow to try and restore, especially in fragmented landscapes, gene flow that would have historically occurred. And we can also look at adaptation. So these are the two components on the right. Thinking about, is there evidence of adaptation? Are there genotypes in the landscape that we could possibly utilise in our restoration plantings to make them resilient to predicted future climates at those restoration sites? Naturally, to use these is really predicated on understanding how genetic variation and adaptation is spread across the landscape. And this is where landscape genomics can really come to the fore. So going back to the example of grey box, so this was much more extensive sampling across the range and looking at patterns of genetic variation and looking for those adaptive variants as well if they were there. So for genetic variation, just generally, this is a PCA. Again, this is a reduced representation data set, and this is a bit over 500 samples across the range. And what we can see, similar to what we found in that smaller subset in the revegetation, is this real isolation by distance pattern, which is more obvious if I turn that graph 90 degrees and you essentially have a map of my sampling distribution. So in terms of revegetation, for this species in particular, we don't have any really strong population structure that we potentially need to be thinking about. And also when we think about what we might consider a provenance, what we might consider local, what we might consider non-local, and therefore how broadly across the landscape we might want to sample, we can actually sample up to a couple of 100 kilometres away and still be within a quite similar gene pool. So these measures can give us an idea of the scale of what is provenance and therefore where we might be wanting to collect in terms of either capturing local variation or alternate variation from other areas of the distribution. Thinking about that adaptive component, we have all new tools and new analyses coming out to look for genomic signatures of adaptation. And for this work, I focused on 10 variables that were predicted to change in southeastern Australia and so are therefore potentially going to be important for future adaptation of this species. And what we find is that, yes, there was evidence of adaptation in Microcarpa. We found a number of significant SNP associations with several of these climate variables that we looked at. Certainly temperature was one of those, as we see in many species, especially those across a latitudinal gradient, as grey box is. Also precipitation, what was interesting was that it was winter and summer precipitation that seemed to be more important or that we found more associations with, which when we think about it from an ecological point of view makes sense. It's not the amount of rain that falls in a year, it's when it falls. And certainly for eucalypts in this area, that summer rainfall is really, really critical for establishment in the first year. So following spring germination, these areas can get quite hot summers. So that summer rainfall is really important to help them establish a taproot, establish their root system to set themselves up and be resilient going forward. When we looked at these genomic variants as well across the genome, we find that they are genome-wide. So as is being found much more broadly, adaptation to climate here is certainly polygenic, it's certainly genome-wide. It's genome-wide. And so between these two, what we have is information on not only the presence of variation, but also some of the key drivers that might be important that we might be wanting to think about and utilise within restoration plantings. So coming back to our restoration strategies that we might have and thinking about those two genetic components of adaptation and genetic diversity, I like to describe landscape genomics as being able to guide where and how widely we should source seed to capture evolutionary potential. So that where is thinking about where in the landscape do we see adaptive variation? What are the gradients? Where are the locations? Where could we potentially source genotypes that may be more resilient to predicted future climates at a particular restoration site? And that how widely is thinking about how broadly do we still want to collect across the landscape, given climate is not going to be the only thing that changes. There are going to be potentially changes in biotic interactions, competitive species, pest species coming in. So needing to also think about maintaining that broad genetic base to keep resilience and provide that adaptability to whatever other challenges may be coming their way. So I think restoration is a really nice case study of how genomics can really help understand variation and be applied to conservation actions. In this second part of the talk, I'm going to shift a bit to how we can use genomics to predict, mostly in natural systems, but again, this thinking could be applied to restoration. So one of the other challenges that we've got more broadly in conservation is trying to predict the capacity for species and systems to respond to change, and therefore to try and get an idea of how vulnerable or resilient they may be. And so as we're growing this number of landscape genomic studies, we're getting this growing body of knowledge about adaptive genomic variation that is related to climate. And complementary to that, more and more studies are starting to use that knowledge to see if we can use genomics to predict change and predict how potentially vulnerable populations may be. So building on the landscape genomic results that I just showed for grey box, I wanted to look at this by looking at what is the change that may be required for those genomic variants that were associated with, in this case, temperature. So each of those points is a particular genomic variant. This is for a population down the cool end of grey box distribution. And what I had is I could measure the current allele frequency, we know that, on the x-axis, but then using the associations that I identified between those SNPs and mean annual temperature, I then calculated what the potential allele frequency change may need to be to match the predicted 2.2 degree change predicted by 2070 in this area, and that's on the y-axis. So we can get an idea of the current state of a population and what that change might be if it's going to match these associations that we have found. And so this is a growing area, and there are a number of people doing a lot of work on this, Christian being one of them, and my approach follows very closely Christian's approach of the risk of non-adaptiveness, looking at what that change in allele frequency is over time. Others are using things like generalized dissimilarity modelling. So this is an example from Megan Supple's work in Eucalyptus melliodora, which also occurs across southeastern Australia. And that's using genome-wide variation and associations between that genome-wide variation, climate, and using that whole genome to predict forward to see what that change may be and identify areas where there may be high or lower change and therefore potentially higher vulnerability to climate change. What all of these have in common is using that genotype climate association that we've identified across space to predict the potential future change that might be required to match projected climate change over time, and using that amount of change as a proxy for vulnerability. I'll just put a little note here as well, that all of this also builds on adaptation from standing variation. So another thing that's really common to all of these landscape genomic studies is that adaptation seems to be associated with shifts in allele frequency rather than the presence or absence of particular variants, which opens up the potential for adaptation from standing variation and thinking about what that change may be and how quickly that change could occur. So this is a really exciting new area, and I find it really fascinating. And like most things in early days, though, there are some really exciting opportunities and prospects, but we also have a number of knowledge gaps. And there were a couple of really great reviews that came out earlier this year touching on those. And what I'm interested in and what I'm going to focus on today is the rate of change. So we can predict an amount of change. So in the case of the grey block example, it was sort of 0.1 to 0.24 projected change. But how feasible is that change? And therefore, how vulnerable may a population be? So there's been a lot of work done on this in, model organisms and really short-lived organisms, looking at what the change may be over hundreds of generations. But I was really interested in looking at what that change might be over a much, much shorter period of time, given that trees don't have hundreds of generations to adapt. And so I'm doing this in two parts. The first part is working with some simulation studies. And so I will preface with that infamous quote that all models are wrong, but some are helpful. And so the purpose of this modelling is not to come up with a definitive answer of precisely how much change we might see in trees, but rather to explore, is there a sensible parameter space where we can see changes that match the predictions, and what parameters most influence this rate of change and therefore might help or hinder genetic change over time. And this piece of work I'm doing in collaboration with Sally Aiken, which has just been fabulous and I've been really delighted to do, as well as Ian McLaughlin, whose PhD work really underpins all the data going into this model. I'll also give a shout out to Jason Bragg as well at the Royal Botanic Gardens in Sydney, who's helping with this modelling, as well as Suzanne Prober inside CSIRO. So it's a really amazing team that I have to work with on this component of the project. And this is working with a species outside of Australia that I've actually never even seen at this point in time, the lodgepole pine. So these occur across the west coast of North America. And within Sally's group and from Ian's work, we have incredible information on how genotype relates to climate, as well as phenotype, and how climate and phenotype relate. So we have that great full triangle of climate, phenotype, and genotype that we can really use to parameterize this model. And so we're looking at a couple of traits. One is growth cessation and cold injury. Given these are competing traits, that capacity to have longer growing seasons, but then that's weighed against the potential for cold injury. One other thing that I will comment about lodgepole pine as well, and certainly the way we're modelling it, is they have serotinous cones, and so they have stand replacing fires. So in one particular stand, you only have one generation of trees. Across a forest, you may have more, but we don't have overlapping generations in this species. So we have a flush of recruitment, selection, and then a fire replacing with a new generation. So looking at some of these models and trying to look at how quickly could genetic variation change over time. And so here we're looking at a couple of different situations. So a couple of different RCP scenarios. Both of these are assuming that we have climate mitigation actions that cause a plateauing of the changes in climate after a period of time. We're also looking at what the effect of, say, high fire frequency and low fire frequency may contribute to these changes. And so what I just want you to take away from this is that in both situations, the genetic variation of the simulated populations, so the light colour lines and the shading, lag behind the theoretical optimum climate given the associations that we have. So they're the darker lines. Under the RCP 4.5 scenario, over time, those stands manage to catch up given the plateauing of climate change in those situations. However, we don't see this in the RCP 8.5 scenario at all. They lag a considerable distance behind the changing climate. I've also got a line there because I think it's really interesting in that 8.5 scenario that the optimum genetic variation actually starts to push up against a frequency of 1, which means that all of those adaptive loci are going to be hitting fixation, which naturally at that point, something else is going to have to come into play, either whether it be different loci or different mechanisms once all those loci are at fixation. Another thing to note here is that those higher fire frequencies enabled more rapid change, as you may expect, given that causes greater generation turnover. More generations, there's more capacity for change. But it should be noted, whilst this is potentially helpful for the trees, that obviously has flow-on consequences for the rest of the ecosystem. So certainly more rapid fires more generally may not be the whole solution here. Looking at the magnitude of change per generation, to start thinking back to those predictions, the grey here are just the neutral loci that we had, where you see not a lot of change and a little bit of change due to drift. And the 4 different colours are those 4 different scenarios that we're currently looking at, so the 2 different RCPs and the high and low fire. Really, all I want you to take away from this is per generation, the average allele frequency change is quite low. We're sort of talking about up to 0.03. So if we're thinking about some of those bigger predicted changes, it's possibly going to take a number of generations for those to be realised. This is still a work in progress. So hopefully we're going to dig into this a little bit more to explore some more parameters and get a better handle on what might shift that, that change per generation. To complement this simulation study though, I wanted to look at some empirical data as well. And so this is coming back to Australia and looking at the beautiful Eucalyptus pauciflora, subspecies pauciflora that we have down here in Tasmania. And so despite trees not being really amenable to multi-generation experiments, I really wanted to look at and ask, what is the allele frequency in the progeny or the seedling cohort compared to the maternal population? And therefore, what's that potential change over a single generation? And can we get some sort of handle on what that may be? And so for this, what I did was collected seed from a natural population, grew those seedlings up, and then split them into control and stress plots. Those stress treatment plots, in our case, was a drought treatment, so we stopped watering them. I'm not necessarily looking at loci associated with drought per se. I was just wanting a stress treatment that would create differential death. So we would have survivors following a selection experiment, and we could look at the genetic variation in the survivors of those and compare those to the controls and the natural population. So really here, what I'm looking at is how much change do we see in the controls, which might be just how much variation do we have in the seedling cohort generally, and what is the change following that selection? What is the change in those survivors, and how does that compare to the natural population? So for this, I've used whole genome pool-seq to investigate those 2 pools as well as the wild population. And so to address that first question, are the cohorts different? Do we see a change just even after that one small selection experiment? And the short answer is yes. So this is a PCA using over 800,000 genome-wide SNPs. And what we see is that the survivors from the drought are more different from the wild than we see in the control plots. So our treatment and that selection, even at that really short time frame, has had an effect, and the survivors are genetically different from the wild and the control plots. Thinking back to wanting to drill down though to what is the magnitude of change that we might see, I looked at just those loci that were consistently and significantly different between control and drought plots across all our 4 replicates. So loci that are potentially associated with that selection pressure that we have applied. And when we look at those and look at the range of change in those, we find that there is quite a range, but again, overall, it tends to be fairly low. So the median is about 0.05, and most of the changes that we see are less than 0.1. We do have some higher changes. However, there aren't very many, so they may not be very frequent or may not have a high probability of occurring. If we then think about comparing back to what are the predictions that we're starting to make in trees and how do these compare to these sorts of numbers, just using the data from the grey box example as just an exemplar of some of the predicted changes. For changes associated with precipitation, we actually saw quite small changes in grey box. And these are actually well within the range of the changes that we're seeing here from that maternal tree population down into the seedling survivors after a selection event, suggesting that maybe some of those changes would be possible in our natural populations in a fairly short period of time. The changes associated with temperature, however, were much higher. And a lot of these stretch right into the top end of the changes that we saw, suggesting that it would possibly take multiple generations to see those changes, and they're possibly going to take, therefore, more time, especially when we think about, even though eucalypts can reproduce quite early compared to some forest trees, so at about 10 years of age, we don't have recruitment necessarily every year. And the other factor, certainly with these eucalypts, is that we do actually have overlapping generations here. So these seedlings are actually going to go into the standing variation that is already present. And so from here, there's, again, this is sort of some early data, and some more work I want to do is thinking about what are the consequences of those seedlings being added to standing variation, what happens if there was some selection in that standing variation, and therefore sort of what's the absolute change in the whole population when you have a mix of both seedlings and adults. And so, as I mentioned, this is a bit of early days, and this is a bit of early data for both of these. But I think what it's really trying to get at is, you know, what is this capacity for change? And it's suggesting that there is some capacity for change over short timeframes, but we're still needing to tease apart a little bit more what that change may be, how that may link to empirical changes that we're actually seeing in natural populations, as well as how that relates to fitness and adaptability under changing conditions. And once we start to put all of those pieces together, I think it will be a really exciting time to really understand how we can use genomic predictions effectively to understand vulnerability to climate change, and how we can use that to help guide conservation decisions. And so with that, I will just wrap up, bringing back to the start and thinking about restoration and all the big goals and the big challenges that we have ahead in restoration and conservation. And yeah, just that I think genomics has a really amazing place in this space. And I think we're still learning what genomics can tell us. And I think these couple of examples highlight that as we think about how can genomics really help us understand how we capture genetic variation in restoration, monitor how well we do, how it can guide where we should be collecting, as well as then trying to predict how vulnerable or resilient a population is going to be, and how we can potentially use genomics to get a measure of that. So we have these incredibly rich data sets, which I think have so much potential. And obviously we still have a bit of work to go in really teasing apart that and understanding it. But I think that provides really amazing opportunities going forward to guide, these really big challenges and these really big investments that are going into protecting our natural systems and restoring our natural systems. With that, I would just like to say thank you all so much to everyone who's tuned in. Thank you again to those who are watching the recording. Feel free, please, to reach out at any time with any questions or to discuss anything further, especially those who aren't here on the chat. I'm always really keen to talk to other people who are working in this area and asking similar questions. There's certainly a lot of brain power here that can come together to really tackle these really, what I think are super interesting and really exciting questions. And I'd just like to acknowledge the amazing colleagues I have who have contributed to the work I presented today, as well as a shout out to many others who I work with and in this space and have had the pleasure of working with for quite some time. With that, I will say thank you and hand back to you, Mariana.
Thank you, Rebecca. This was really inspiring. It's great to see how genomics can help us handle the questions that we have when we are doing either enrichment plantings or restorations. Really, really interesting. And I'm sure we have some questions already. Does anybody want to ask a question?
Hi, I have a question myself. So
please, Santi.
thank you so much, Rebecca, for your talk. It was really a really good one. Thank you so much. Wanted just to ask you about, you have mentioned it that well, there is some work to go to connect the predictions with adaptability and fitness. So and that's a special concern I have is exactly that. It's about validation of these predictions that are mostly based on genomic data, and then about ways of how can we use common gardens or forest inventories to validate these predictions. And I would like to know your opinion on that. Which ways do you think would be better to connect this genomic prediction with real phenotypes and real life? So
yeah, that's a really great question, and I don't think there's a silver bullet, but what comes to mind is certainly the point that you raise of how these predictions play out in reality. What I've presented there is rate of change, but the other question that's always at the back of my mind as well is that are the loci that were associated with historic adaptation, as in the ones that we're identifying in a genotype environment association studies, are they going to be the ones that are important in the future? So, which parts of the genome are we looking at? And I think to answer that is really going to be those longer term studies. So either potentially plantings, and they can be common garden trials, or even say utilising restoration plantings. I know there's discussions here in Australia about how we can think about incorporating some experimental thinking into revegetation planting, so we can look at them long term and see what the changes are and see how these predictions do play out. So I think it's going to be monitoring that change over time in experimental plots. So who is surviving over time, especially those extreme events. If we have an extreme event, who has survived through that extreme events, understanding they're going to be some of the key pressure points. I also love to think that there is some capacity to possibly look in natural populations, and I know there have been a couple of studies that have come out recently, and Christian has certainly been involved in one of them, to look at what is the natural change we're seeing in systems where we have the capacity to look at older standing trees and then look at recruits coming through. What are the changes that we are seeing in those, and monitor that over time. So look, I think there's a number of different ways we can come at it, but they all come down to exactly as you say, it's that monitoring over time. It's having that prediction and then actually seeing what happens over time and feeding that back in to really understand how they fare.
Thank you. Thank you so much.
Thank you, Rebecca. And I just want to say that keep raising hands, and please, you can also use the chat to ask the questions. And I see Bruno. Please, Bruno.
Yes, thank you, Mariana. Good morning, good afternoon. Yeah, thank you very much for the talk. I think that geneticists in general are mostly optimistic people, and you show that actually, yes, variation occurs and changes can happen. But I think what you also show, and tell me if I'm wrong, is that that is true up to a certain point. Then if we follow some scenarios like the 8.5, well, we're actually pretty much doomed in terms of how much genetics can help adaptation, or diversity can help adaptation. And I was wondering if you have an idea of where things start to not work out. We at a global level, the COP26 again pushed for the maximum of global warming of 2 degrees since 1850. It's better 1.5. I wonder if for us geneticists and with the models that you used, if there is such a threshold over which actually genetics cannot, or genetic diversity cannot help survival, and can you see this, or is that too early or impossible to do?
Yeah, so it comes back to this, I suppose, thinking about that certainly in that model, the changes in genetic variation that I'm looking at are associations that we've identified between phenotype and climate. So there's one aspect of there may be other loci that perhaps come into play that could aid adaptation. In terms of that change overall, yeah, I don't have a graph at hand, but certainly there is, as you mentioned, a particular amount of change at which the associations that we have between, say, genotype and climate or genotype and phenotype, if we follow the predicted changes, they hit the point of fixation, or the predictions are beyond. So loci have become fixed. I don't have an exact answer as to, you know, what would happen beyond that, but that's just really flagged to me as you're saying that there is somewhere in the system there is an upper limit. So there's going to be allele frequency changes potentially aren't going to be the be all and end all of adaptation, and whether that's because there are going to be different loci that are going to come into play or different mechanisms that come into play. Yeah, I don't think I have the answer to that, except, as you say, to flag that there's a point at which, given our current knowledge, given the models and associations we have, we hit fixation. And so allele frequency change no longer becomes an option for future adaptation beyond that. Yeah, I'm not sure if that quite answered your question, but it's a big question to ask.
Yeah, thanks. Yeah, I think it does, and for me it's important because I think as geneticists, as I said, we're very optimistic that diversity is the option. But as you say, well, you know, one generation may be enough for some traits, but not. It also depends on the age of regeneration. But I think for communicating with other fields and with practitioners, I think we need to be pretty clear that it's not the absolute answer, and genetics will not be the answer after certain thresholds, which, as you say, is not completely obvious to pinpoint. But well, for me, the 2 degrees threshold seems to be a good one. But yeah, thanks.
Okay, and I have a question myself. You know, we are facing a lot of, I mean, we had large-scale disasters, and we are facing restorations or enrichment planting in part of our forest. And I was wondering if you would give any opinion on which of these 4 different sourcing strategies might be the most effective. I mean, I know that you cannot say it for Europe, but at least you can elaborate that on the eucalyptus and give us a little bit more explanation on how to see those different strategies.
Yeah, look, that's a really great question, and also goes to these strategies that have been proposed given, you know, our scientific understanding of adaptive variation and genetic variation. But one of the, similar to the genetic predictions, one of the really interesting things, and one of the areas that we're currently moving into, is how do these actually play out in reality? So how do these different alternative strategies work in different scenarios? So that's data that really needs to be built up. So we can sort of have this discussion from, yeah, this sort of empirical science understanding, but how that will play out in reality is work that is currently happening and that we're trying to understand. And so it can also, sorry, this is going to be one of those answers where it's like, you know, totally depends on context, which I'm not sure.
Of course, of course.
You know, some of the considerations that come into play is, you know, if you're looking at something that is, that passively doesn't have really strong adaptation across environmental gradients, then maybe you're thinking about capturing good genetic diversity more generally, or mimicking patterns of gene flow. Whereas if you do have adaptation and adaptation associated with projected changes, then maybe that could be utilised in those restoration plantings. And certainly something we have in Australia, I'm not quite sure in your situation, but certainly across the landscapes that I mentioned that I've been working on, they're quite highly fragmented. So one of the other issues with sticking with just local provenancing is sometimes you can be left with just a couple of trees here and a couple of trees there, which you can imagine the genetic diversity in those is going to be pretty low. So it's sort of balancing those as well, going, okay, how much further afield can we collect to make sure we get good genetic diversity, even if it isn't quite specifically the really local diversity that we want. But really the shorter answer to your longer question is that we're really needing to understand and really test some of these strategies to see how they play out in different scenarios for different species, because some might be better, say, for annuals and things with short generation times, as opposed to trees, which are obviously much longer generation times, and in any individual is in the landscape a lot longer. So I don't think I've given you an exact answer there, except, yeah.
But you confirm that what we really need to do is try and then keep monitoring what happens and then feed that back into our models and see, right?
Yeah, absolutely.
Yeah, that's great. And I see Kostya having also a question. Please, Kostya.
Hi, everybody. Can you hear me?
Yes.
Thank you, Mariana, and thank you, Rebecca, for nice presentation, very interesting data. But more data become available now, saying us that epigenetic modifications are probably more important in response to adaptation to environmental stress factors. So, and especially those which are stable and can be inherited transgenerationally. So what do you think about this, that we have to take epigenetic modification into account, pay more attention, and how would you tackle this problem? What is your opinion on this?
Of tackling the problem of epigenetic variation or other?
Right.
Yeah.
Because it helps genetic variation. It helps to survive. It's a fast response to environmental change, while genetic changes are very slow, as you demonstrated. But epigenetic modification is a fast response. It's a first defense line, I would say, for trees especially. So what do you think about this?
Yeah, absolutely. There's going to be, I mean, what I've focused on has been DNA sequence, mostly because that's sort of what I've been working on. It's sort of a natural progression through. But certainly, yeah, when starting to explore those changes and what might be the capacity of those changes really does get you thinking about the fact that there are going to be other things that come into play, and epigenetics is one of those. I think most definitely that's growing. It's certainly not a field I have a lot of knowledge of, but it's certainly exactly as you say, those DNA sequences and those genomic sequence changes are going to be quite slow. And so, yeah, if you are thinking in those rapid timeframes or those extreme events, epigenetics, I think, is likely to have a role, and exploring what its role may be and how it complements DNA sequence changes and how those two come together, I think will be a really interesting area to really get a better understanding and sort of come back to the point that Bruno made, you know, what is the adaptive potential, because there are going to be other mechanisms that are going to come into play. So how do these come together to help that adaptability going forward? So yeah, I think it's a really interesting area, and it'll be really interesting, yeah, to see how it helps and what it can contribute in terms of resilience and adaptability going forward.
Thank you. Great. Good luck with your study.
Thank you.
Okay. Please, Christian.
Yes, I have a comment in the question, actually. I have a comment to Santi's first question. I mean, validation is really important, and they are coming now. So actually, there's one study by Matthew Fitzpatrick with Provenance trials. There's another one that Brandon Lind showed at the EVOLTREE conference, and there's also simulation studies coming now. And in the Provenance trial studies, they just kind of predict the genomic offset or the vulnerability to the provenance, the location of the provenance, and it worked quite well, at least better than purely climatic distances or purely genetic distances. So it seems to somehow work. We don't know in which cases and which not, but there's a positive sign. Then my question is, what do you think? I mean, a bit similar probably to Kostya. I mean, there's other factors influencing these things. If the real frequency changes are not big enough to face the future, do you know anything about migration or about has the distribution of your tree species that you looked at, has it changed during climate change? What can you say about that factor, migration?
Yeah, that's a good question. I'm not sure about what migration has potentially happened. One of the really big challenges, certainly for grey box, the Eucalyptus microcarpa, and a lot of the species that are in that agricultural space, is the not capacity to move. There's a sentence there that I can construct better. Because of the agricultural matrix that they're in, the capacity to move across the landscape is much, much lower or inhibited, or they just simply can't. They're going to be cleared because of that production landscape. So there's and one of the issues is fragmentation and the fact that there isn't landscape to move through. One of the other slight challenges we have in Australia is we have really low topographic relief. So we don't have, whilst we do have some mountains, we don't have those really sharp elevational gradients that certainly for microcarpa, we do have some species that live on the mountains, and so they have that potential. But microcarpa is much more on the flat. And so therefore, there isn't a lot of capacity to change quite quickly in terms of going up elevational gradients. And so it's actually quite large distances it would have to cover to, say, get into a different temperature zone. So coming back to your question, I'm not sure of precise measures of move, whether distributions have already changed or if anyone's measured what that capacity would be to change. But I certainly know there are a number of factors there which are just limiting the physical movement of those trees through the landscape, which is going to make that more challenging.
So then you are trying to restore them by collecting seed from other populations. I wonder from how many trees are you collecting, in order, you show the differences in the diversity, but how much big of a sample of mother trees do you have when you carry out these plantings?
That's a really great question. And that was one I was hoping to uncover in my study, as well as the review that we did of the published literature. And that was one of the really big knowledge gaps, was that, well, certainly in the published literature, it's not mentioned. I'm not sure if people know behind the scenes that we unfortunately can't get access to that information. But from my experiences, when I was looking for those sites, people could usually tell me the year it was planted, maybe where they sourced the material from, but in terms of number of mothers, number of populations that were sampled, we just really didn't have that information. So that's some information that would just be so powerful and be so informative to really understand what might influence genetic variation, because I think as you're sort of going towards, if we would expect at some point that increasing the number of mothers at a site, increasing the number of sites, there would be some increase in diversity, and perhaps that plateaus at some point. So we can, there is an optimal point in that sampling effort, but we certainly didn't have access to that data and weren't able to find that, and therefore weren't able to provide those recommendations. So I think going forward, especially given all the efforts people are putting in, that would just be something, if some of those metrics could be captured, would just create such a powerful data set to help that monitoring to understand what is the influence of collecting from more mothers or more populations. Does that have an impact on diversity? Is there an optimum point? Does it vary between life history types, again, short-lived annuals versus trees, and some of that data? But yeah, certainly we weren't able to find those answers in the study.
Yeah, that's a shame, because we would really need hard data on those numbers, especially with so many reforestation planned in the future. So we really make it resilient. That would be very nice to have. I mean, in Europe, we usually recommend collect from 25 trees, but also there is, we could do more on the data there to really get to the right numbers per species. So
yeah.
Are there any questions? Any more questions? Bruno, please.
Yes, thank you. I was wondering if, how how large is the use of planting in forestry in Australia? Let's say for the species you showed us, is it in Europe? It depends on the country. Some countries use massively planting, or like British Columbia also, but others rely a lot more on natural regeneration and so on standing genetic diversity. I was wondering in Australia, how does it work? Is it more plantations? Is it more natural regeneration?
Yeah, that's actually a really, really good question. I focused on plantings for my study, so I'm less familiar with natural regeneration. So I think I'm suspecting actually there are a couple of other people who might be able to answer this question better than I. Yeah, so look, I won't put any hard and fast on that because I'm not sure, but certainly there is a lot of effort in planting back, certainly in agricultural areas. We're talking about areas that don't have a lot of standing trees for that natural regeneration to occur. So certainly for grey box, I think that predominantly focuses on planting back into the environment. I'm not sure about other ecosystems which may potentially rely to some extent on natural regeneration as well, but a lot of my experience has been more in those planted landscapes. Yeah.
Okay. I mean, we are also, it's 10 minutes past 10. So if there are any questions, this is the time to, any questions for Rebecca? This is the last chance now to take them. And I'm sure if you come up with another question, Rebecca will be very happy to answer you by mail or another, or a Zoom or a phone call or a Zoom meeting individually. Yeah. And with that, Rebecca, I really thank you because your talk was great and inspiring, and thank you for all the explanations and the answers. Really, really great. And I haven't read all your papers, but I'm sure I will enjoy reading them now. I will search for them immediately after the talk. Thank you really much. And before we close, yes, thank you, Christian. I would like to invite you for the next talk in this third series of the webinars. Next week, standard time, 4:30 on Wednesday, Emily Latch will be talking about genomic data, about how genomic data improves wildlife conservation. And there is still time to register if you haven't. And I hope we will see as many of you there as well. And with that, I would also like to thank you, the audience, for the questions, for listening, and for making links with Australia. And hopefully we will find some collaborations after this also between Australia and Europe. So thank you, everyone. And thank you to the Christian, Stephen, Santi, and Felix for organizing these talks. Thank you all, and see you next time. See you next Wednesday.
Thank you.
Thank you. Thank you.
Bye-bye.
Thank you, Mariana.
Bye.
Everybody.
Bye.
Thank you, Rebecca. Have a good night, I guess.
Thank you.